Hello, thanks for coming to my virtual talk. My name is Kelly. I'm a PhD student at Stanford in Anshul Kandaji's lab, and this talk is about a model I've developed called ProCapNet, in which we study the cis-regulatory syntax of transcription initiation using deep learning. So why are we doing this? Well, in recent years, there's been a lot of really awesome progress modeling the role of DNA sequence in various components of gene regulation. So you may know BPNet, a model that predicts TF binding directly from sequence. There are lots of others. We've also had similar success with things like accessibility being modeled directly from sequence, and even 3D genome conformation from Zhang Zhao's work called ORCA. And yet, even though these models work really well, whenever we try sequence to direct gene expression models, those tend to struggle. Models like Informer are not quite at the performance ability that we would like them to be, and they're also often not very interpretable. So why is this? This is the hypothesis that I'd like to propose. When we're trying to go direct from DNA sequence to the transcriptome with a model, we have these variables that we're imagining are being learned from sequence by those models, things like TF binding, chromatin state, and 3D contacts. We know that deep learning models can learn these, so why can't they just go ahead and predict the transcriptome? Well, the reason is because the biology is really a little bit more complicated. So all of those variables impact transcription initiation, but when you measure the transcriptome with something like direct RNA-seq or CAGE or Rampage, you're also measuring the outcome of things like elongation rates, RNA splicing, other RNA processing, and the stability of those transcripts, which affects how long they stick around in the cell. So we're proposing that one way to sort of compromise here is to model specifically transcription initiation as opposed to the steady state transcriptome, and then hopefully we can recover some of the really great model performance that we see when we model some of the components on the left there. So introducing ProCapNet, this is a sequence to transcription initiation model using ProCap data. ProCap measures capped nascent RNAs at base resolution, so we have really precise measures of where the TSSs are and also the amount of RNA that's being nascently transcribed. This captures both stable and unstable RNAs, which is what makes it different from something like CAGE and Rampage as well. We capture eRNAs just as well as promoter RNAs. The ProCapNet is a BPNet-like model. It has two tasks, notably. The first is what we call the counts task, which predicts just the overall ProCap signal in a given locus, sort of akin to intrinsic promoter strength in a way. And then the second task measures the distribution of TSS positioning per strand. So this is the base resolution aspect of the model. So overall, this model makes fairly accurate predictions. On the left is how the counts task performs. A Pearson correlation of 0.71 is pretty good, given that this model doesn't know enhancer gene linking. This is only looking at local promoter sequence. And on the right, I'm showing a lower bound and an upper bound in the extreme lines. And the middle bound is how well we do. We're closer to pseudo-replicates than to an average profile lower bound in terms of profile performance. So that's also decent. To show you some specific example. So the top row here is the observed ProCap at a given locus. You can see that it's more dispersed than you might expect TSSs to be. This is one of the differences between ProCap and CAGE. The second line is how well the model performs. You can see that the two lines kind of match each other. And something that we can do with these deep learning models is we can interpret them using something called DeepShap, which assigns scores to all of the input bases in the sequence that we fed into the model to make this prediction. If we do that and then zoom in so that we can see what bases are highlighted by these scores, we can see known promoter TF motifs that pop out, such as NFY, the BRE, or the SP1 motif, and NRF1. We can run then a clustering algorithm called Modisco, which basically summarizes those high-scoring motifs into a set of clustered motifs. And this is the set that we get out. So you can see that we find two forms of the initiator sequence. We find two forms of the Tata box, we find the TCT motif, and we find several other promoter motifs that we would expect to see. Model predictions are pretty accurate at these motifs. And because we have two tasks in this model, we can differentiate between how each of these motifs contributes to the profile task versus the counts task. And we can see that there are differences. For example, ETTS primarily contributes to the counts task, and so is more of an activator than a positioner of the pick, for example. So we can look at where these motifs fall relative to summits. For example, the INRs tend to be right at the summits in either unidirectional peaks up top and bidirectional peaks on below. We can look at the strand specificity too and confirm that INRs are very, very strand specific. We can look at where the Tata box positions itself, and as we expect, it's about 30 base pairs upstream of either of the summits and also orientation specific. And we can look at something like NFY, which is a third example where we see really interesting positioning relative to the summits. So one of the hypotheses we wanted to test with this model was if we look at enhancers versus promoters, how does our model interpretation differ? So we do see that in enhancers, overall, we're getting a lot fewer motifs and also fewer unique motifs than we are in promoters. If we try to figure out exactly which motifs that is, it tends to be NRF1 and the SP1 binding sites that are most enriched in promoters relative to enhancers. And then AP1 is the motif that tends to be most enriched in enhancers relative to promoters, which aligns with current understanding of biology. We can also look at motif strength in enhancers versus promoters, and there are significant differences for most of our motifs. In particular, for most motifs that are enriched in promoters, we also see that they have stronger motif strengths on average in promoters, which makes sense. We've done one more test besides this, which is that we've trained a model on only promoters and asked how well it generalizes to enhancers relative to the normal ProCapNet, and we have seen that it generalizes equally well. This evidence and more has led us to believe that what's going on at enhancers for transcription initiation is conserved with respect to the cis-regulatory syntax compared to what's going on at promoters. We can also do some really interesting in silico experiments with this model. So here I'm showing you the MIC promoter, and I'm going to mutate some motifs and then ask the model how its prediction changes. You can see that the observed and the predicted for the original sequence match really well. If I then look at this Tata motif on the right and ablate it by just replacing it with different sequence and then have the model make a new prediction, we can see that the TSS on the right just totally disappears. If I do the same thing with the INR that's over there, we get most of a loss of that peak, but not the same as what happens with the Tata box. And I can do the same with the Tata box over on the left for the upstream TSS and compare how it affects the upstream versus the downstream TSS. And finally, I can add new initiator sequences and see how that impacts what's going on. This is just a proof of concept as to what sorts of things we could do with variant effect interpretation with this type of model. And finally, we can do really cool motif spacing simulations with this model. So here I'm taking the Tata box and the initiator, and I'm putting them in sequences at various spacings. And you can see that right there, there's a really cool synergistic effect where the Tata box is exactly 30 base pairs upstream of the INR, and there's a high super additive effect that spacing has in terms of the amount of initiation that comes out of where the INR is. To watch it again. So in summary, we have a model, ProCapNet, that can accurately predict transcription initiation at base resolution using only the local DNA sequence as input. It's interpretable, it's not enormous, and it learns the expected promoter TF motifs that you would want it to learn. We use this to look at what's going on at enhancer RNA and find a conserved cis-regulatory logic there compared to what's going on at promoters. And we can perform all sorts of really cool in silico mutational experiments to figure out what the rules are that this model has learned, such as motif spacing, motif orientation, etc. Thanks for listening.